Hello, everyone, and welcome to today's webinar, a case study of protein sample prep strategies for membrane proteins. Our presenter today is Dr. Barbara Kaboord, Senior Research and Development Manager, Protein Biology, Thermo Fisher Scientific. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, before we begin, I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by using that Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Barbara Kaboord. Dr. Kaboord is the Senior Research and Development Manager of the Protein Preparation Group at Thermo Fisher Scientific, where her team is responsible for the development of numerous protein extraction, stabilization, purification, and cleanup products. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Kaboord, you may now begin your presentation.
Thank you for the kind introduction, and welcome to all of you who are joining us online for this presentation. To start off, let's look at a high-level overview of today's talk, where I'll start with a description of the 5 steps, what to consider as you're strategizing your protein prep procedure, followed by a case study in which I'll discuss our approach to prepping G-protein coupled receptors, or GPCRs, for activity and structural analyses. At the end, there will be some time reserved for questions. So what are the 5 steps in prepping a protein? To prep a protein from cultured cells or tissue of any type of in vitro analysis, the process starts with making a lysate or extract. This can be either a whole cell lysate to solubilize all proteins, or some type of subcellular fractionation where different cellular compartments such as membrane, nuclei, or other organelles are differentially solubilized with detergents or separated by differential centrifugation. During the extraction process, cellular compartment integrity is often compromised, potentially exposing your protein of interest to proteases and phosphatases that are normally sequestered elsewhere. An essential component then of any lysis buffer formulation is a mixture of protease and or phosphatase inhibitors to preserve the integrity of the solubilized proteins, as well as preserving any labile post-translational modifications that may occur naturally in situ. Second, purification or enrichment of some kind is usually desired. This can be done in a variety of formats involving chromatography resins and columns, or magnetic beads and magnetic separators. Affinity purification is usually the preferred approach, and a variety of ligands are available to facilitate this, and I'll discuss this in more detail shortly. Classical chromatography resins, such as strong cation or strong anion, are also vital to many purification schemes. So once you have your purified protein fraction, some sort of cleanup is beneficial. Both dialysis devices and desalting resins are frequently used to remove unwanted chromatography salts and small molecules that might have been used to competitively elute protein from affinity supports, as well as to exchange proteins into a buffer more suitable for long-term storage. Protein concentrators and diafiltration can also be used for this purpose, in addition to their more obvious function of concentrating proteins for more stable storage. Step 4 of the protein prep workflow is quantitation. After all that work, how much protein did you yield from your purification process? Multiple protein assays exist to help answer this question, BCA and Kumasi protein assays being the most popular. Lastly, the 5th step, detection, is important to the workflow since it is here that you determine qualitatively how well your protein prep worked. SDS page, Western blotting, and or mass spectrometry are often used to evaluate protein size integrity, and verify the identity of the protein isolated, as well as the presence or identity of any remaining contaminants. So, how do you start? The protein preparation procedure you choose is largely dictated by the nature of the sample, by the cellular location of the protein you're isolating, how much protein you need to perform your assays, and most importantly, the identity of those downstream assays and applications. So what do you want to do with your protein in the end? Applications such as Western blotting or mass spectrometry do not necessarily require a lot of protein, nor do they require your protein to be functionally active, in most cases anyway, while applications like ELISAs, immunoprecipitations, reporter assays, and activity assays require the protein to be in its native conformation and stabilized. The next few slides will walk through some of the factors to consider when designing your protein prep workflow, such as choosing an expression system, what, if any, epitope tag to design into your starting DNA construct, how to best extract or solubilize your protein of interest, do you need to enrich or purify that protein, and if you do need to purify, then at what scale and what supports are available. These factors and decisions will then be highlighted in the context of our project that aims to solubilize, stabilize, and purify GPCRs for activity and structural biology applications. To start off, let's back up and take a quick look at the basic model systems for overexpressing proteins. The assumption here is that you're overexpressing a recombinant protein, not isolating an endogenously expressed protein. So the options are, starting at the top, a cell-free system, which is essentially an open system requiring no live cultures. It consists of a specialized lysate made from E. coli, wheat germ, rabbit reticulocyte, or mammalian cultured cell sources, and is often used in a high throughput format and is good for expressing small amounts of proteins. Its challenge is that large-scale expression is usually much more difficult. The workhorse of expression system is bacteria, and that's because this system is nice in that it's scalable, it's relatively low cost, it requires simple culture conditions, and the expression time is usually short in duration. There may be some issues, though, with expressing certain proteins in bacteria. Some proteins are not soluble in this system. There are minimal post-translational modifications added to the expressed proteins in bacteria, and some mammalian proteins may just not express well. So another option is to go to a yeast system, which is also low cost and simple. It does have the advantage of having able to conduct eukaryotic protein processing, but fermentations are required to produce very high yields, and growth requirements may need to be optimized. Insect cell cultures are also low cost. They have an advantage of that the post-translational modifications, or PTMs, are similar to mammalian systems, and they're good to express proteins that are normally toxic to mammalian cells. The culturing conditions are a little bit more demanding, and the PTMs are not quite identical to mammalian systems. So the gold standard then really is, for expressing mammalian proteins anyway, is a mammalian expression system, as this will provide the highest level of correct post-translational modifications and provide the highest probability of obtaining fully functional human proteins. The downside is that the culturing conditions are more demanding and that high yields are best achieved with suspension cultures. So if we look at this next slide, I've outlined 3 different commercial expression systems that have been adapted to grow to high density in suspension, eliminating the need for the end user to do this, and optimized to obtain maximal yield from mammalian and insect cultured cells. Human 293 cell system was developed especially for end users performing structure function studies that require native protein folding and post-translational modifications. CHO, or Chinese hamster ovary cells, are the workhorse of biopharma, as many biologics are expressed in CHO. Both systems generate high protein yields of up to 1 to 3 grams per liter. More recently, an optimized insect cell, or SF system, has been developed to provide cost-effective platforms for academicians as well as biopharma doing vaccine development. So once you've identified or chosen your expression system, is there any type of affinity handle that you would like to engineer into your construct to facilitate purification? So if we start at the top, IMAC tags are the most popular and relatively easy to use and are low cost. His tags are pretty much ubiquitous in the protein purification world. However, their one drawback is that the affinity of this interaction is lower than the other tag choices, and background contamination is usually higher as there are more metal interacting proteins and lysates. However, this is still the most popular choice for purification tag. GST has a higher affinity, but it is also a larger, more bulky addition to any protein. Its advantage is that it can improve solubility of proteins, especially in E. coli systems, as can another tag, MBP or maltose binding protein. Small peptide tags have the advantage of a small size, like the His tags, but the affinity with their corresponding antibody supports is orders of magnitude higher, enabling much cleaner protein preps. The disadvantage is cost of these resins and minimal reuse and regeneration. However, these tags are really popular for immunoprecipitations and are often used for larger scale purifications when IMAC resin just doesn't cut it with respect to purity. In addition, the flag tag has a built-in enterokinase cleavage site, which provides another strategy for eluting from columns and makes the tag removal from the N-terminal tag proteins easy. So since His tags are popular, I wanted to take a brief amount of time to discuss some options for the corresponding IMAC resins used to purify them. The schematic on the left shows the inverse relationship with protein yield and purity as a function of metal ion. Nickel is the most popular version of IMAC, as it strikes a good balance between purity and yield. If purity is a major concern, then cobalt supports are best, with the caveat of potentially compromising a bit on yield. Also, the number of interaction points between the chelator on the resin and the metal also influences purity. Lower number of coordination sites result in higher protein capacities, but are also a bit dirtier. NTA is the most popular chelator, as it provides good capacity while being fairly resistant to the effects of other chemical entities in the lysate and binding buffer. The pentadentate chelator, shown on the bottom here, is highly specialized in that it is resistant to reducing agents, EDTA, and other amino acids or metabolites in cell culture media, but has the drawback of a lower binding capacity in most situations. So one other tag that I wanted to comment on is the flag tag, or the DYK-D4K epitope tag. This is an extremely popular tag as it's small, minimizing any potential interference with the overexpressed protein, and it interacts strongly with its corresponding immobilized antibody resin, resulting in very high purity protein preparations. It's a great tag, and while I highly recommend the use of this tag, I wanted to draw your attention to a potential issue when using this tag with membrane protein expression constructs, especially since the case study presented later in this talk features membrane receptors. There are a couple of reports in the literature where flag tag was used to tag transmembrane and secreted proteins, but the authors were not able to efficiently purify these proteins. And the problem found was that the tyrosine residue in the sequence was sulfated in the Golgi, especially with N-terminal tag placement. This sulfation modification of the tyrosine interferes with the recognition of the epitope tag by some of the anti-flag antibodies available. So if you use the flag epitope tag on overexpressed membrane proteins and can't successfully purify it on anti-flag supports, is likely not a problem with the chromatography resin or the beads. So it's best to be safe by choosing a different epitope tag for membrane or secreted proteins. So choosing your extraction strategy is also dependent largely on downstream applications, and these may influence the choice. For example, if you're characterizing proteins, modifying, labeling them, or determining their structure, purification is usually required. So starting with by preparing a simple whole cell lysate extraction is usually the easiest starting point. Other applications, such as protein interaction assays, monitoring disease states, tracking biomarkers, or studying cell signaling cascades, may be accomplished without purification, but may benefit from some type of enrichment to help increase the effective concentration of the desired protein. Enrichment is often accomplished by differential detergent solubilization of cell compartments using specialized cell fractionation buffers. So most purification strategies begin with a whole cell lysate buffer, optimized for the model organism used to express the protein. There are lysate buffers optimized for mammalian, bacterial, or yeast cells, maximizing the total amount of protein solubilized while still being gentle enough to maintain native structure and function. And some of these are listed in the middle column of the table on the right. Alternatively, there are buffers and formulations that enable nuclear cytoplasmic fractionation, membrane protein enrichment, or other subcell fractionation, providing methods to enrich for protein classes or particular cellular locations. The extraction buffer that I will discuss later in this talk that was used to solubilize GPCRs has characteristics of both types of lysate buffers, and that it results in essentially the production of a whole cell lysate, but it is specialized as well in that it is able to stabilize a certain subset of proteins, the GPCRs. So getting back to our 5 steps, if purification is required, what scale of purification is needed, and what chromatography support is best for the job? As you can see, there are multiple options at any scale. Magnetic beads and small-scale spin columns are best for nanogram to microgram quantities, for example, to do IPs and pull-downs. Magnetic agarose can be used as an automatable alternative to small agarose columns for a milligram to about a gram of protein. Larger research scale purifications requiring FPLC columns work best with cross-linked agarose or superflow resins. Lastly, porous resins are a type of bioprocess media that enable high flow rate, large volume bioprocess scale purifications, but I won't really discuss any more of that type of resin at length in this talk. If automated purification is desired, there are 2 main types of magnetic supports: traditional low micron size magnetic particles and larger high capacity magnetic agarose, the latter being porous and providing a much larger binding capacity. Because of this higher capacity, magnetic agarose can be used to automate small scale purifications and can also be used to screen expression levels during optimization experiments. So moving on, almost everyone prefers the speed and efficiency of affinity purification over classical chromatography. Many of these ligands, especially those to purify tagged recombinant proteins, are available on both chromatography resins as well as magnetic beads. Classical resins, such as cation and anion exchange resins, hydrophobic interaction resins, etc., still have their place when purifying endogenous proteins, which have no overexpression tag, or as a polishing step after affinity chromatography to maximize purity by removing small trace levels of contaminants. So now we'll apply some of these fundamentals to solubilizing and purifying GPCRs to be used in downstream activity assays and cryo-EM structural studies. So I probably don't need to tell many in this audience the importance of membrane proteins, but as is listed here, they're involved in a variety of cellular functions, ranging from intercellular junctions to cell-cell recognition, cell attachment, enzymatic activity, and signal transduction. And thus they are critical for normal cell homeostasis and any aberrant signaling that can have serious consequences and manifest as a variety of disease states. G-protein coupled receptors, or GPCRs, are 7 alpha helical transmembrane domain proteins that play important roles in transmitting cell signals across the plasma membrane. There are over 1,000 different GPCRs encoded by the human genome, making them the largest class of membrane proteins. They serve as effective drug targets for a variety of genetic and immune system disorders, as shown here on the right. And they serve as potential biomarkers and therapeutic targets for cancer treatment. There are currently at least 475 drugs approved by the FDA that act on over 100 unique GPCRs. One of the main challenges in GPCR research is the ability to efficiently extract the receptor from the membrane without compromising the structural and functional integrity of the receptor in order to study it. Isolating and studying GPCRs presents many challenges. Their complexity makes them hard to clone and express. They are embedded in the plasma membrane, making extraction difficult. And efficient extraction often requires harsh conditions that result in denatured or unstable proteins and low yields. Any kind of activity assay then usually needs to be done right away. Buffer components that may help to stabilize GPCRs outside of the cell membrane often are found to interfere with downstream analyses. For example, detergents and glycerol are not compatible with mass spec or structural studies such as cryo-EM. This case study that follows is a work in progress. We don't have all the answers yet, but we're making significant progress in addressing some of these challenges, especially in extracting and stabilizing GPCRs, maintaining function throughout the initial purification process, and working to minimize contaminants and substances that may interfere downstream. Most of the emphasis will be on the first three steps of the protein prep process: extraction and preservation, purification, and cleanup. There are several strategies to extract GPCRs from both cells and tissues. Subcellular fractionation uses one or more of the properties of membrane proteins, such as density, surface, size, and shape, to separate them based on differential centrifugation in media of high viscosity, such as sucrose or sorbitol. Aqueous polymer two-phase system is a liquid-liquid fractionation technique. High salt or high pH buffers can be used to dissociate proteins from the cell membrane, but can also damage protein integrity. So most of these traditional protocols are laborious, time-consuming, and require extensive ultracentrifugation equipment, as well as extra steps to stabilize the target protein. For these reasons, we decided to focus on detergent extraction. And like the components of biological membranes, detergents have both hydrophobic associating properties and polar head groups that allow the dispersion of hydrophobic compounds into solution. At high concentrations, detergent monomers aggregate into structures called micelles. And working at or above the critical micelle concentration, or CMC, promotes detergent monomers to form colloidal aggregates or micelles that can sequester hydrophobic membrane proteins into their structures, effectively keeping them in solution. For obvious reasons, we wanted to avoid using denaturing detergents, such as SDS, as these would not be ideal for maintenance of both structure and function of GPCRs. So we decided to focus on non-denaturing detergents for extraction. After testing a variety of detergents, including several of those listed here in this table, we determined that a mixture of non-ionic and zwitterionic detergent gave the best results when extracting GPCRs. Not only do we want to fully solubilize the GPCRs, but they also need to maintain their native form and function afterwards. As integral membrane proteins, they are generally unstable when removed from their membrane environment. Different strategies for stabilization include proteoliposomes, which are systems that mimic lipid membranes and incorporate the protein within; nanodiscs, which are nanometer scale planar discs of bilipid layer stabilized by an encircling scaffold protein. Next, the SMALP approach is similar to nanodiscs in that it involves the spontaneous encapsulation of proteins from a membrane into a wrapped nanoscale particle, but instead of a protein scaffold, styrene maleic acid wraps the protein lipid particles. Amphipols are short amphipathic polymers designed to absorb tightly onto the hydrophobic surface of the transmembrane proteins and covers them with a thin interfacial layer of surfactant. And lastly, the inclusion of sterols stabilizes by interacting directly with the GPCR encapsulated within a micelle. The approach commonly found in the literature is to first extract the GPCRs from the membrane, followed by a separate step to stabilize the extracted receptor. We determined, however, that the receptor is better stabilized if both the extraction and the stabilization happen concurrently. Because of this, we developed an all-in-one formulation that contains all of the components needed to extract stable and functional GPCRs. So not only did we want an all-in-one formulation, but we also wanted to optimize the extraction protocol to be fast and efficient. So this slide outlines the basic protocol. So if you follow the numerical sequence, starting with your sample, whether that be tissue or cells, you wash your sample in cold PBS, spin it down, discard the wash, and then resuspend the resulting pellet in GPCR extraction and stabilization reagent. Suspend it and incubate it for 30 minutes at 4 degrees. Following the incubation, the cell debris is pelleted by centrifugation at 16,000 times G for 20 minutes. And from there, you'll remove the supernatant containing the functional receptor and proceed to either purifying your tagged receptor from the whole cell lysate or taking your extract directly into your downstream application. So some of the common downstream applications or assays for GPCRs include, if you start at the top of the slide, immunoassays, ligand binding assays, surface plasmon resonance, functional assays such as GTB gamma acid exchange assays, structural studies such as NMR, X-ray crystallography or cryo-EM, and thermal shift assays. Initially, we wanted to show that we were able to efficiently solubilize endogenously expressed GPCRs from both culture cells and tissues. So the Western blot on the left shows 3 different GPCRs extracted from 2 different cell lines using 4 different extraction strategies. These approaches included 2 different detergent-free membrane preparations, A and B, and 2 detergent-based extraction buffers. Both detergent approaches were more efficient at solubilizing the various GPCRs if you compare the soluble or S labeled lane to the P or pellet lane. These are more efficient at solubilizing than the detergent-free methods, with the optimized GPCR reagent, which is sample C, being equal to or better than the harsher RIPA buffer in sample D. Similar results were observed with the liver and brain tissue samples on the right. Next, we wanted to demonstrate that what we isolated was functionally active, and we did this by initially using radioligand binding assays to show specific ligand binding to the receptor. Extracted GPCRs were incubated with tritiated ligand specific for the respective GPCR. Unbound or free ligand was removed using a small molecule removal resin, and GPCR samples in the flow-through were counted in a liquid scintillation counter. Our first trial was with the M3 receptor. In the left panel, the Western blot shows 99% of the receptor expressed in CHO K1 cells was solubilized using the GPCR extraction reagent, both with and without the stabilizing component. This shows that the stabilizer itself does not aid in extraction, but looking at the activity data in the middle panel, you can see that the initial binding activity is about 2-fold higher when the stabilizer is present during the initial extraction. So if you look at the dark bars in the middle top graph. The presence of the stabilizer is absolutely required to maintain active receptor during storage at 4 degrees over 10 days, or at up to 1 month at -20 C. You can see that in this case, storage of those receptors remain stable and retain greater than 75% of the initial activity. This reagent adds significant flexibility with respect to time and allows for follow-up experiments that would normally require a new preparation on a day for each assay. Additional receptor activity assays were performed to show that the GPCR extraction and stabilization reagent was able to efficiently extract different GPCRs from stable expression cell lines as well as tissue. The right panel shows the stabilizing benefits of the reagent for the ADORA 2A or A2AR activity during prolonged storage at 4C. So for up to 2 weeks, you can see that activity was successfully maintained. A new and promising tool in membrane research is the detergent-free solubilization of membrane proteins by styrene maleic acid copolymers, or SMAs. These amphipathic molecules are able to solubilize lipid bilayers in the form of nanodiscs that are bound by the polymer. So membrane proteins can be directly extracted from the cells in a water-soluble form while conserving a patch of native membrane around them. So this slide will show 2 protocols. So on our left is our current protocol, and on the right we have a sample SMALP, SMALP protocol, in which we tried both the lipodisc lipid formulation and an amphipol reagent to surround and stabilize the GPCR, which is then wrapped by the styrene maleic acid assembly. So both methods are relatively straightforward, with the SMALP protocol being slightly more involved with respect to reagents and time required. But what do the results have to say? So we wanted to compare both the solubilization and the stabilization properties of the SMALP and GPCR reagent approaches. The extraction results for 2 receptors, the glutamate receptor and the beta-2AR receptor, are shown in the Western blots on the top left panel. Extraction efficiency was similar or better with the GPCR reagent compared to either SMALP method. Adenosine receptor 2A, or ADORA2A, was solubilized from mouse brain tissue with the whole cell lysate method and the GPCR reagent, or the SMALP method using either lipodisc 3.1 or amphipol for polymer stabilization. Radioligand binding assays show that the extracts made with the GPCR extraction and stabilization reagent have greater than 2-fold specific activity from endogenously expressed A2AR when compared to the SMALP preps. So in this case, all methods were able to extract, all methods were able to stabilize, but the GPCR reagent performed at least twice as well. So moving on now to purifying these extracted GPCRs. We've previously validated that the reagent was able to efficiently extract and stabilize the activity. Ultimately, we want to extract, purify, and stabilize for downstream structural analysis by cryo-EM. So we created a construct to express a GPCR, either the A2AR or the CB2 receptor, as a fusion with GFP, followed by a his tag on the C terminus. Since these GPCRs are mammalian in origin, we chose to express in the XB293 system to better our chances of generating correctly folded, highly active protein. Next, we then diluted the extract 1 to 10 in the nickel IMAC binding buffer. We wanted to keep the GPCR reagent components present at about 1 times the CMC concentration in order to keep the membrane protein soluble and stable throughout the purification, but minimize any potential detergent interferences. Following elution, imidazole was removed using a Ziba desalting column and the GPCR analyzed by SDS page, quantified by protein assay, and analyzed for activity by radioligand binding assay. We were initially surprised when we checked the extraction efficiency of the overexpressed GPCRs in the higher expressing XB293 system. While the GPCR reagent normally solubilized greater than 90% of the endogenous or lower level expressed GPCRs, we solubilized a significantly lower percentage when the GPCR was in a more highly expressed system. We tried adding a bit of mechanical disruption to enhance the action of the extraction reagent and found that gentle down homogenization or sonication increased the amount of GPCR in the soluble fraction, which is shown on the left panel. However, activity assays showed that while more protein was solubilized with sonication, the highest activity was obtained with a gentler down mechanical disruption, and that data is shown on the right. In all methods, there was still GPCR in the insoluble fraction, and we hypothesized that the membrane fluidity or composition is slightly altered with higher levels of expression, and that makes the membrane more resistant to solubilization. Thus, additional mechanical disruption may be required in these situations. It is also possible that the overexpressed GPCR that remains insoluble may not be material that you actually want to use, as it may be improperly folded or aggregated. So taking the solubilized GPCR and purifying it over nickel NTA agarose or magnetic agarose, according to the workflow described in a previous slide, we show that we can purify the Cb2-GFP fusion. The Western blot on the right confirms the identity and the size of the isolated construct, and that it was efficiently captured and eluted from the IMAC purification support. If you look at the lanes marked E for elution. The silver stain gel on the left is a bit busier to look at and harder to see the target protein, but it does show that while the Cb2 is present in the elution, another purification step will be required to remove residual contaminants. Either a dual affinity tag will be used in the future or an ion exchange polishing step. Similar results were seen with the adenosine receptor construct. Western blots on the right show good binding and elution from the nickel NTA magnetic agarose beads, whether you're detecting with the A2AR antibody itself or an antibody against the GFP or the His tag. The silver stain on the left shows overall purity and the residual contaminants that remain. However, the real question is that before further optimizing the purification, we wanted to check the ligand binding activity of the A2AR receptor after the nickel IMAC purification. The data shows that the adenosine binding activity was retained throughout the purification, which is good, and that the A2AR specific binding activity was significantly enhanced in the purified and eluted fraction. We are confident that we can maintain active protein throughout the purification workflow as long as we retain low concentration of the initial GPCR extraction and stabilization components throughout the course of the purification. So to wrap up, what have we talked about? First of all, we talked a little bit about protein preparation workflows and how they're largely influenced by the number and nature of the downstream applications and analyses that you want to perform. There are multiple techniques for solubilizing stable GPCRs. Optimized detergents extraction can improve the entire protein prep workflow for endogenous receptors from extraction through detection. For overexpressed receptors, some gentle means of mechanical disruption is often required to augment detergent extraction methods. And using a combination of detergent and stabilizer during extraction greatly increases the stability of the extracted receptors and allows for extended storage at either 4 degrees or -20. This combination of detergent and stabilizer is required throughout the purification process to maintain receptor function. And GPCR solubilized using the detergent stabilizer extraction method with the Pierce GPCR extraction stabilization reagent yield more functional receptors compared to other polymer techniques such as SMALT and amphifols. Lastly, I just wanted to reiterate the 5 steps in the protein preparation workflow, starting with extraction preservation, purification, cleanup, quantitation, and detection. And I've highlighted a few of the critical reagents and tools used to generate the data that I showed here today. So I'd like to thank my scientists and collaborators who helped generate the samples, the overexpression samples, as well as the extraction and purification data that you've seen here today. And at this point, I'd be happy to take any questions you may have.
Thank you, Dr. Kaboord, for that excellent presentation. As you mentioned, it is now time for our live Q&A portion of our webinar. Now to our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We already have some great questions coming in. Dr. Kaboord, let's begin with this one. Why did you choose XC293 to express the GPCR constructs?
Okay, it's a good question. So, as I mentioned, the GPCRs were mammalian in origin, so we wanted to match the expression system to that. But also, what researchers in the field have been finding is that as you extract the GPCRs, a little bit of the endogenous lipids from the originating membrane come along for the ride as you either create micelles or some of these nanodisk particles, and that those endogenous lipids are actually critical for function. So the best way to stack the deck and hope that you isolate active receptors is to express it in the native system, as well as give it a chance to bring some native lipids along for better activity.
Thank you. Our next question, is there a particular reason why you added GFP into the CB2 and the A2AR constructs?
So yes and no. We had that construct, I think, for another purpose, but we decided to use it because ultimately we wanted to hopefully extract and purify protein that could go into cryo-EM studies. And the larger the protein in that case, the better for just obtaining the required resolution by cryo-EM. So a combination of the GPCR fused to the GFP creates a larger protein molecule, and then combined into the lipid micelle, or the detergent micelle, excuse me, we hope to then create a large enough structure to see successfully by cryo-EM.
Now, Dr. Kaboord, is the background seen on the silver stain gels of the iMac purification normal?
It is. If you have a protein that is really highly expressed, like a soluble protein, and you have a big band on your starting lysate lane, most of that protein then all that protein occupies the space on the affinity resin, okay? And there's not room for a lot of contaminant binding. In the case of the GPCRs, that's, even though they're overexpression systems, they still don't overexpress as dramatically as other soluble proteins. So some of that is just the inherent nature of working with membrane proteins and it being a difficult system, and then some of it is because we probably can adjust that the resin to sample ratio a little bit to maybe decrease the resin and minimize space for nonspecific binding.
Thank you. Now we have time for just a few more. Have you looked at other GPCR activity assays and interaction assays with G proteins, agonists, inverse agonists?
Not yet, but we intend to. To get that credibility in the GPCR field, we know we need to do those assays. We started with probably the easier assay to set up, which is the radioligand binding assay, and so we're kind of screening conditions using that. But ultimately, yes, we want to show that we've isolated and purified active functional GPCR. We will need to go into those assays, and that's something that some of our collaborators within the company are going to help us with.
Thank you. And we'd have to wrap with this final question. How long can you keep the solubilized GPCR stored and stabilized?
So I show data for 1 week at 4C and up to a month at -20C. We have extended those studies. We found that we can't really go much beyond 1.5 to 2 weeks, and I think that may be somewhat receptor dependent when you're storing them at 4 degrees. But I think our latest data is that we can go up to potentially 2 months at -20. So it's not indefinitely, but it is significantly helpful compared to having to isolate and the receptors and doing all your assays same day.
Thank you. And thank you again, Dr. Kaboord, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions. Questions that we did not have time to answer today and those submitted during the on-demand period will be addressed by Dr. Kaboord via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. Hope to see you again soon. Bye-bye.